Yeah, this is Syed from TechInvention, welcoming you to yet another very interesting webinar as part of our knowledge dissemination and series program. For the benefit of all the audiences, for all the audience members who have joined us, EBPMN Emerging Biopharma Manufacturers Network is a pan-global network of biopharma manufacturers and also those aspiring to be manufacturers. We do have members from Latin America, Asia, and Africa. And I can see that a few colleagues have joined us from Latin America. It's quite early in the morning for them, so it's good morning to them. Also from Africa, where it is early afternoon or a bit late in the afternoon. It's a pleasant evening for all of us out here in India. And of course, a little late evening for our colleagues who have joined in from South Asia, Southeast Asia, as well as China. So would like to welcome you all to what promises to be a very insightful webinar. Incidentally, today I have the pleasure of hosting one of our co-founding members, CNBG, which is a subsidiary of Sinopharm, to take us through this webinar. For a short introduction, CNBG Group has more than a century, or rather, to put it straight, 105 years since inception in 2019. What used to be seen as largely a vaccine-oriented company, they have taken giant strides in expanding their portfolio all across. And today they have a presence in biosimilars, in plasma-derived therapeutics, diagnostics, antibody products, and so on. The contributions extend to over 100 countries, partnering with global organizations and supplies to UNICEF to ensure healthcare access worldwide. And to take us through the profile and portfolio of biosimilars and novel biologics at CNBG, we have a very accomplished scientist out there, Dr. Zhang Chi. Let me quickly introduce Dr. Zhang Chi. He's the director of early stage drug development programs at the Shanghai Institute of Biological Products. He holds a degree in Bachelor in Pharmacy from the San Yat-sen University. Thereafter, he's done his Master of Science in Biochemistry from the University of Cambridge. Dr. Zhang then went on to do his PhD in cancer biology from the Institute of Cancer Research at London, and he brings over a decade of experience in drug discovery and development. Prior to his role, he contributed significantly to advancing a first-in-class drug candidate to IND stage during his tenure at Cancer Research UK, London Research Institute. At CNBG, Dr. Zhang leads a highly skilled team dedicated to identifying early-stage drug candidates and driving discovery biology research. His contributions include 16 research publications and 2 PCT patents, and is recognized as a fellow of the Shanghai Society of Biotechnology. Without further ado, would like to leave the floor to Dr. Zhang to take us through his presentation, towards the end of which I'm sure Dr. Zhang will be happy to take some of the questions from the audience and clarify any further aspects that we need to. So Dr. Zhang, welcome to this webinar, and the floor is yours to take over.
Many thanks. To start with, let me try to share my screen. And I hope everyone can see it.
Yes.
Right. So it's a great pleasure to join this EBPMN forum, where we can come together to share insights and forge collaborations in the biopharmaceutical space. So my name is Chi, Dr. Chi Zhang, and I'm honored to represent CNBG today. And over the next few minutes, I will share an overview of our capabilities, our global footprint, and our innovative antibody products that are making a difference in the life of patients worldwide. So let's start with an introduction of our parent company, Sinopharm. So Sinopharm is a global healthcare leader, the first Chinese pharmaceutical company to enter the Fortune 500. So currently ranked 112th. So with 3,000 retail stores, 1,600 subsidiaries, and over 220,000 employees. Sinopharm provides the infrastructure and the reach we need to deliver high-quality biologicals and biosimilars to patients worldwide, which directly supports the EBPMN's mission. Within Sinopharm, China National Biotec Group, which is short for CNBG, focus on innovations in biologics and biosimilars. We connect with 5,000 suppliers, 3,600 distributors, and cover 31 provinces, offering more than 30,000 healthcare products. Through global partnership and R&D investments, CNBG rapidly adapts to evolving healthcare needs, and both at home and internationally. Our extensive manufacturing networks ensures reliable access to critical therapies, and with 30 global standard delivery centers reaching 80% of major Chinese cities, and operations spanning 31 provinces and 268 cities, we swiftly scale productions and distribution. This agility is essential when addressing urgent healthcare challenges and expanding access in low and middle income countries. And as the heart of CNBG's achievement is the Shanghai Institute of Biological Products, or SIBP. The SIBP has 75 years of experience in biopharmaceutical innovation and covering 370,000 square meters. SIBP combines advanced research with large-scale production capability, and over 1,400 skilled employees ensure that we can produce more than 40 million doses annually, driving both public healthcare campaigns and the development of next-generation biologics that can improve patient outcomes around the globe. Now let's look at how SIBP ensures the manufacturing excellence. So to meet with the rising global demand, and SIBP has built a world-class production infrastructure. We operate a 30,000-liter antibody production line that supports a diverse range of therapies. Inside our facility, we use both 2,000-liter single-use bioreactors for flexible batches and a rapid turnaround, as well as 6,000-liter stainless steel bioreactor for large-scale output. And all process adheres to stringent GMP standards, and we have localized critical equipments and the materials for greater efficiency. And this manufacturing strength underpins the commercialization of biosimilars like trastuzumab, rituximab, and bevacizumab, ensuring patients get high quality and affordable treatments. And with these capabilities, SIBP has emerged as a true hub of antibody industrialization, ready to scale productions and remain globally competitive. At SIBP, our pipeline of nearly 20 antibody products addresses major unmet needs. We focus on oncology and tackling common cancers like breast cancer and lung cancer, and also on autoimmune diseases such as arthritis and psoriasis, and also on infectious disease that still threaten many communities. And the three of our biosimilars stand out. The SIBP A01, which is a trastuzumab biosimilar for the HER2-positive breast cancer and the gastric cancers, and the SIBP A02, and rituximab biosimilar targeting NHL, CLL, and autoimmune conditions. And also SIBP A04, a bevacizumab biosimilar for multiple solid tumors, including colorectal and ovarian cancers. And beyond the biosimilars, we are also advancing next-generation treatments through our antibody-drug conjugates. For example, the SIBP A13 targets HER3-positive tumors with a novel payload and cleavable linker, showing potent anti-tumor activity and enhanced binding affinity comparing to competitors. Similarly, SIBP A17 focuses on HER2-positive cancers, offering efficacy on par with DS8201, but with reduced toxicity. These ADCs reflect our expertise in sophisticated linker payload technologies, enabling us to deliver precision-targeted therapies that improve safety and potentially transform outcomes of the patients. And so let me propose the most of our collaborations that could happen at SIBP. So at SIBP, we see collaboration as many ways to accelerate innovations and broaden patient outcomes. First, we welcome partners who wish to license our advanced monoclonal antibodies. Along with these licenses, we offer comprehensive technology transfer that we can even work together to develop new antibody variants that meet your specific needs. Secondly, we provide access to our proprietary linker technology for those interested in creating their own antibody drug conjugates. These arrangements include a full technology transfer, initial training for the R&D team, and ongoing technical support to ensure a smooth and productive partnership. And finally, we also offer opportunities to produce and commercialize our developed ADCs in designated markets. Here, we can support you with technology transfer, production training, and even supply key initial components to help you establish a strong market entry. So our goal is to form partnerships that could drive innovation, streamline local productions, and ultimately make life-changing therapies more accessible in the worldwide. At SIBP, we also recognize that different partners have different needs. So we've developed the flexible collaboration models as well. For example, exclusive regional licensing arrangements that could give you distribution and the management rights in specific territories, while you handle the local clinical trials and the regulatory submissions. We also encourage co-development initiatives where we work hand in hand to create new products or enhance existing ones. By pooling our expertise and the resources, we can accelerate progress and address unmet medical needs more effectively. Additionally, we invite partners to consider licensing or co-developing some of the innovative products emerging from our research pipeline. This approach offers direct access to cutting-edge technologies and the therapies that have a potential to significantly improve patient care. Each of these collaboration models designed to deliver mutual value and bring effectively treatment to patients wherever they may be. So let me start with the more detailed view of our capabilities. So first of all, let me start with the biosimilars. At SIBP, we have built a robust portfolio of biosimilars that reinforce our commitment to quality and affordability. One example is the SIBP A01, trastuzumab biosimilar that has shown therapeutic equivalence to Herceptin and is indicated for HER2-positive breast and metastatic gastric cancers. And another SIBP A02 is a rituximab biosimilar, effectively against a range of conditions, including non-Hodgkin's lymphoma and the CLL. We also offer SIBP A04, a bevacizumab biosimilar equivalent to Avastin, which is used to treat various solid tumors, from the colorectal and lung cancers to ovarian, renal malignancies, as well as hepatocellular carcinoma. So focusing first on the SIBP A01's clinical validation. Our biosimilar SIBP A01 demonstrated clear bioequivalence with Herceptin in clinical trials. On healthy male volunteers, it is just as safe and well tolerated as a reference drug, which is Herceptin, with no notable differences in safety or immunogenicity. This evidence confirms that SIBP A01 matches the established profiles of Herceptin, ensuring that patients can trust its quality and efficacy. And similarly, the SIBP A04's trials confirmed its value. In our phase 1 clinical trials, the SIBP A04 proved bioequivalent to Avastin, and the healthy male volunteers tolerated well, and with no meaningful difference in the safety or immunogenicity comparing to the original drug. And the pharmacokinetic profile aligned closely with Avastin, reflecting the rare and the care and the quality behind the A04's development. In addition to the traditional targets, we also target emerging pathways such as the HER3 with SIBP A03. So SIBP A03 stand out as the first anti-HER3 antibody drug clinically approved in China with full IP ownership. And HER3, unlike other HER family of members, lacks intrinsic tyrosine kinase activity, but plays a pivotal role in tumor growth through interactions with EGFR and HER2. And overexpression of HER3 is common in various solid tumors, including breast, ovarian, prostate, colon, pancreatic, and lung cancers. By specifically targeting HER3, the SIBP A03 offers a novel strategy for tackling these challenging malignancies. And our preclinical trials has shown that SIBP A03 significantly reduces tumor size in xenograft models like FADU, A549, and NCI-N87 without causing substantial weight loss, an indication of its low toxicity. And across the range of the 2 mg per kilogram to 40 mg per kilogram, it maintained an excellent safety profile. And when combined with cetuximab in the head and neck squamous cell carcinoma models, the SIBP A03 achieved an objective response rate of 37.5%. And these results highlight its potential as a safe and effective option for patients with HER3-driven cancers. And beyond oncology, we also address inflammatory conditions through the WIBP adalimumab. This is developed in the Wuhan Institute of Biological Products, a sister institution to the SIBP. The Wuhan Institute of Biological Products has developed this biosimilar to Humira, and that this is known as the WIP adalimumab or Wuhan adalimumab. And the Humira is a well-established therapy that targets TNF-alpha, helping to reduce inflammation associated with various conditions. And our biosimilar, marketed under the name Shen Ailin in China, works the same way. It binds both soluble and the transmembrane TNF-alpha effectively to decrease its activity. And the WIBP adalimumab covers a broad range of indications. And we've also ensured that it can benefit children, and it has been approved use in pediatric diseases. And each vial is available as a 40 mg per 0.3 ml formulation, and we anticipate it to be available in the Chinese market by March 2025. And now let's come to our ADC the ADC innovations in more detail. The SIBP has developed the next generation ADC platform that raised the bars of precision and reliability in the ADC design. So our innovative linker technology improves the solubility and ensure higher conjugation efficiency and enhances overall consistency. And with a cleavable payload linker structure, we have achieved precise drug release and even a bystander effect, which can eliminate the neighboring tumor cells without harming the healthy tissue. And by increasing the hydrophilicity of our linkers, we streamline the conjugation process and reduce unwanted homopolymers and boost manufacturing quality. And this platform has already produced multiple ADC clinical candidates, with an average drug to antibody ratio of 7 to 8, and showing remarkable versatility. And among them, the SIBP A13 stand out as a particularly promising therapy. The SIBPA13 embodies our advanced ADC approach and pairing a humanized anti-HER3 antibody with a cleavable linker and a DXD-based topoisomerase 1 inhibitor. And this design ensures that the cytotoxic payload is directed precisely to HER3 positive cells and leading to the efficient internalization and the target killing of tumor cells. So in preclinical studies, and SIBPA13 demonstrated superior binding affinity and greater cytotoxicity than U3-1402. This is a notable ADC in the field. It also proved effective in xenograft models in the EGFR negative cancers, retaining its potency even when EGFR-driven resistance was present. And the early phase 1 clinical data has reinforced the SIBPA13's potential. The dose escalation phase reached the 5 mg per kilogram without any dose-limiting toxicities, indicating a very favorable safety profile for SIBPA13. And at 2 mg per kilogram dose, one patient has experienced a reduction of tumor diameter from 77.3 millimeter to 57.8 millimeter just after 3 treatments. And these findings suggest that SIBPA13 can offer the highly targeted, effective therapy for patients with HER3-driven malignancies, and representing a significant step towards a forward in ADC treatment strategies. And similarly, SIBPA17 illustrates our capacity to tackle HER2-positive cancers. And SIBP-A13 is a HER2 targeting ADC that targeting the HER2-positive cancers with the same sophisticated linker payload technology. This approach delivers cytotoxic payload directly to the tumor, significantly reducing the risk of off-target effects. And our preclinical study has shown the minimal hematological toxicity and positioning A13 as a safer alternative compared to many existing ADCs. Given its strong preclinical performance, we believe SIBPA17 has a great potential to benefit patients living with HER2-driven cancers. And in our ongoing phase 1 clinical trial, the SIBPA17 continues to show both safety and the promise dose and the promise. And the dose escalation has progressed smoothly and reached 4 mg per kilogram without any dose-limiting toxicities, which indicates a favorable safety profile. Perhaps not more notably, and the early efficacy signals are encouraging. And the one patient at the 2 mg per kilogram dose level has experienced a reduction in tumor size from 69 millimeter down to 33 millimeter just after just 2 treatments, given every 3 weeks. And this finding supports the idea that SIBPA17 could become a transformative therapy for patients with HER2-positive malignancies, solidifying SIBP's position at the forefront of ADC innovation. And to wrap up, so today we have explored how Sinopharm and the CNBG through the SIBP and the WIBP are broadening global access to biopharmaceuticals. Our scalable manufacturing, diverse pipelines of biosimilars, and also novel ADCs and the collaboration models highlight our commitment to addressing unmet clinical needs, especially in the emerging markets. We invite our partners all over the world that can advance innovation, local capacity, and the patient outcomes. And thank you for your time, and I look forward to work together to transform the healthcare for those who need it most. Thank you.
Well, thanks a lot, Dr. Chi. That was a very lucid articulation of the kind of work that's happening at CNBG. You have a very interesting portfolio of 20 plus candidates, flexible business models. I'm sure you'll do exceptionally well all across not only the global south, but also global north, where you have plans. We do have an audience of 50 plus, which have remained with us from the beginning till now. And I'm sure there will be some questions which I would like to invite uh, the audience to put up in the chat box out here. I have a few questions as the audience prepares to put some questions in the chat out here. The first question is, with several products targeting the HER2 and the PDL1 pathways, how is your company addressing redundancy and competition within its own pipeline as well as against established market players?
Right. We do recognize that the redundancy can arise when multiple therapies targeting similar pathways, such as the HER2 or HER3. And to address this, we carefully differentiate our products by focusing on specific patient subpopulations and also on unique mechanisms of actions or novel combination therapies. So this includes the refining the antibody engineering or optimizing drug to antibody ratios for ADC products, or exploring the synergy with other agents for other drugs. So we also continuously evaluate the clinical data to prioritize the most promising candidates. So by maintaining a robust and data-driven approach, and embracing the partnerships, we ensure that each product offers a distinct clinical advantage. That's how we differentiate our therapeutic approach.
Yeah, I think the HER3 is a very interesting concept to pursue, and I think that would add to the differentiation and innovation into your portfolio that you're pursuing as compared to the HER2 PDL1, which is more of a biosimilar approach. I mean, makes tremendous sense though. There are a couple of folks in the audience who have significant expertise in IP, as I'm able to see them. So my next question to you is around the IP side. In the context of your collaboration opportunities, how do you manage intellectual property, IP protection, and technology transfer, especially when working with external partners?
Right. We approach collaborators with clear and structured framework for IP management. Before any partnership begins, we will negotiate terms that define each party's rights and responsibilities concerning the technology and the know-how and other joint developed IPs. So this includes the establishing a detailed licensing license agreements and confidentiality provisions and clear milestones for the technology transfer. And through this collaboration, we also provide the technical support and the training to ensure effectively knowledge sharing and the transfer. So our goal is to foster a trust that values all the partners.
No, makes tremendous sense because while you do file patents through the PCT route, I suppose a lot of local prowess and proficiency is required to protect IP. So it does make tremendous sense to have the shared values and working closely with your partners. We have one question from Dr. Tiago. The question in the chat is, does your main differentiation strategy on developing the linker for ADC? So is that the main differentiation strategy?
Right. So our linker technology is designed to be cleaved under very specific physiological conditions that found predominantly in the tumor microenvironment, such as certain pH levels or the presence of particular enzymes. So this linker technology is developed by a rational design of the amino acid sequences. that will foster the release of the toxic payload in a very cancer specific way. We actually have the full IP ownership of these linker sequence that not only allows the specific delivery of the payload, but also eased the process of manufacturing.
Great. So I think that's the reason why you're also willing to work with other companies on taking this technology globally. Yeah. It's a good strategy to pursue. There can be no discussion on biosimilars, novel biologics without a question on regulatory. So we do have the next question on regulatory. The question is, given the global regulatory landscape, how are you navigating the regulatory challenges for the approval of your biosimilars and ADCs in key markets like the US and Europe?
Right. So we approach regulatory challenges by maintaining open dialogues with agencies early in the development process. And our strategy includes aligning our clinical trial designs and manufacturing processes with the global standards, and also proactively seeking the scientific advice whenever possible. And for biosimilars and ADCs, we follow well-established guidelines to demonstrate similarity, safety, and efficacy, while investigating in robust quality control and pharmacovigilance systems. So by staying informed of evolving regulatory requirements and engaging in timely consultations with the regulatory authorities, we are trying to ensure that our products that meets the highest standard for approval.
Okay, thanks. The next question is around the PKPD, which is a big challenge when it comes to the large molecules and biologics. So
Right.
this question is specifically around pharmacokinetics. So what strategies are being implemented to enhance the pharmacokinetic properties of your antibody drugs, particularly for those in the early development stage?
Right. That is a very good question. Actually, this is the question that we've been pursuing for the long time. So we are taking a multifaceted approach to optimize the PK profile from the very earliest development stages. So first, we refine antibody engineering to improve the antibody stability, the half-life, and the tissue penetration. And this might include the FC fragment modifications or glycoengineering. And we also carefully select the linkers and the payloads that maintain stability in circulation and release for those ADC products. And additionally, we employed advanced analytical methods and modeling to predict the PK behavior and guide the formulation decisions.
Perfect. Doctor, we have a question in the chat box from Dr. Meena Adel. How can Sinopharm, that is CNBG, benefit from global and regional initiatives that promote and support local production in Africa?
Yes, that is a very good question. Actually, the CNBG's operations in Africa is one of the main facet of collaborations that we have been focusing on. And what we are seeking is to bring about affordability for those monoclonal antibody and ADC drugs, including biosimilars and innovative ADC drugs. So in this way, as I mentioned early on, we can benefit from through our partnership. We can we can benefit from the technology transfer and the licensing royalties, so on and so forth. And actually the most of collaboration is quite flexible, which is on a case by case basis.
Sure. So to put it straight, CNBG is quite open to collaborations, flexible
Yes.
business models, and Africa is a key market out there. And I mean, those who are interested should for sure get in touch with CNBG for the same. There's one more question from Bhakti. The question is, do you have any products in the pipeline where you are keen to collaborate in global clinical trials, global multicentric trials?
Right. We do have this, especially in the field of ADCs. As I presented early on, the SIBP A13 and A17, which are the HER2 ADC and the HER3 ADC. Because we have achieved the full protections of these the intellectual property and expertise from the very early on development of these drugs, that we are very keen to collaborate, especially with those clinical experts, to conduct multi-centered investigations and the clinical application of these prototypes.
Perfect. So I think yes, even for global filings today, it's important to have the global multicentric trial. So glad that you're looking at that as a strategy for your early stage discovery drugs.
Yes.
We've had quite a few questions in the chat box, and quite a few questions which we had decided to specifically get into. Thanks, Dr. Cheap. I suppose you've put across a very nice overview of the kind of development that's happening out there, and all-encompassing strategy, the kind of business models, and you've handled the questions so well. Since there are no further questions in the chat, we would like to thank you and the team CNBG for setting up this opportunity to present CNBG and Sinopharm through the EBPMN network. I'm sure the audience has quite a few more questions that may come up. We will be glad to take up those questions through EBPMN, or also you can directly talk to CNBG. There was an email in the slide of Dr. Chi where you can write to. Otherwise, we'll also happy to support you from EBPMN. I would like to thank specifically my colleagues Simran and Sonali, who have worked closely with the CNBG team in setting up this webinar. Hence, we have an audience of 50 plus which have participated from Latin America, Asia, and Africa. Would like to thank you all for a nice rest of the week, and look forward to joining you in the next session of EBPMN series. Thank you.